Welcome to part 2 of our reproductive systems lecture. We're going to talk about the structures and a little bit about the functions of the male reproductive system. This is going to match page 546 to 549 in your textbook. All right, we're going to just introduce the structures and then we'll talk about them individually. And this is very similar to the picture that's on your textbook or the figure that's on your textbook on page 547. As we said in lecture 1, the gonads of the male system are the testes. And remember, these are the primary sex organs. They're in charge of making the gametes. In this case, the male gametes are sperm. They're also in charge of making the sex hormones, so in this case, mainly testosterone. Then there's a duct system, which leads from the gonads out to the external part of the body, and there's going to be several different parts of this pathway. And this is the way that the semen is delivered. And then there's accessory glands, and these make the semen. And lastly, there's the external genitalia, the things that surround these internal structures and help with delivery of the sperm. So they house the testes, and again, they aid in the delivery. All right, so first we're going to talk about the primary male sex organs, which are the testes. They have two types of secretions. The exocrine secretion, and remember exo means external, so this is going to make this and then send it elsewhere, is the sperm. The endocrine secretion, endo meaning internal, meaning it's going to secrete this and keep it, is testosterone, which is the male sex hormone. Then there's the tunica albuginea, and this should say fibrous capsule here. This is the part that surrounds, here you can see it in white, that surrounds the internal workings of the testes. In the interstitial cells, meaning the cells that are in the tissue, so one of its jobs is to surround, but then in its actual cells, it has these cells that produce the actual testosterone. Then we have the seminiferous tubules. That's in yellow here, and you can see there's quite a bit of them. They're coiled up, and then they dip back, and they're sort of divided into these lobules, and this is what's going to actually make the sperm. And once the sperm is made, it's going to lead through this part, okay, the rete testes, and that's going to lead to the beginning of the duct system. We'll talk about that in a few minutes. Oh, sorry. Okay, so the testes have the two main jobs: make the sperm and make the testosterone. And then sperm, again, exocrine means to send somewhere else, are going to leave. Testosterone is going to stay within the male body to regulate various different things that make males males. Okay, so we have the capsule here, the tunica that surrounds it and that makes the testosterone, and it's surrounding these seminiferous tubules, which are making the sperm, which are then leading out to the duct system. Okay, let's talk about the duct system. The first portion of the duct system is the epididymis. So after the sperm leaves the seminiferous tubules, it's going to go into what was on that last figure, sort of in pink, the epididymis. Again, it's very, very, very coiled. So let's go back for a second. Okay, here, just like the seminiferous tubules, we've got a lot of coiling. It's got to work all the way through these coils, all the way down these coils, all the way back up these coils. And the reason for that is to allow enough time for the immature sperm to mature. So when it's all said and done, this is about 6 meters long, which takes about 20 days for the full trip for the sperm to make. Okay, and by that time, they should have matured. Also, the walls of the epididymis are designed to contract, so that helps to move the sperm along. Next, they make it to the ductus vas deferens. Sometimes you just hear this called the vas deferens, and what this actually means is carry away. Okay, so this is designed to do exactly that. This is actually moving upwards, so it has to move against gravity. So the vas deferens is going to lead up out of the scrotum. It's going to go through what's called the spermatic cord, and that's going to lead up along with blood vessels and nerves back up into the abdominal cavity. So we can see here, here's the epididymis. They've made their way through all those coils over 20 days. They've matured. Now they're leaving the scrotum, carrying up here. It's going to wrap all the way over the bladder and back down here and end to meet the urethra to leave. Okay, at the very end, it's going to widen into something called an ampule, and that's right here. It actually isn't labeled here, but it widens right before it leads through the prostate. So that's sort of a staging area for the sperm until ejaculation actually occurs. The whole length of this epididymis has thick walls of smooth muscle, and we've seen this before in a lot of the previous systems that we've talked about. This is for peristalsis, so this is going to actually move all of that sperm along, just like we saw in our abdominal system, just like we saw in our urinary system, and that allows movement against gravity. Okay, the last part of the duct system is the urethra, which leads out of the penis, out to the external environment. And unlike in the female system, this actually is the same pathway as urine. Okay, so it carries both sperm and urine, but never at the same time. So when ejaculation occurs, and this is part of the reason that that staging area is right here, right before the prostate and right next to the exit of the bladder. When ejaculation occurs and sperm is going to be moving through the urethra, the internal sphincter of that bladder closes, that muscle closes up so that no urine can leave. So it's never carrying them at the same time. Okay, the accessory glands. So these are going to be things that are aiding in the making of semen and sometimes also in the secretion of other types of hormones, are the seminal vesicles. And then we also have the prostate gland and the bulbourethral glands, which we'll talk about momentarily. The seminal vesicles secrete seminal fluid. The prostate gland secretes sort of a milky white fluid. And then the bulbourethral glands secrete a thick, clear mucus. The first two, the seminal fluid and the milky fluid, are going to be added to the sperm, and those three things together are what makes semen. This thick, clear mucus gets secreted before the semen for two reasons. It's going to cleanse the urethra, and it's also going to serve as a lubricant. So it basically goes first to clean out any urine or anything else that might be in the urethra, and to serve as a lubricant. Okay, so this is not a structure, but we need to talk about briefly what is actually contained in semen and why, and then we'll move on to the external structures. So semen is sperm plus those two other secretions that we talked about from the seminal vesicles and from the prostate gland. And all this together aids in the delivery of the sperm. So sperm, you can see a picture here, very, very, very magnified. Sperm are cells that are streamlined for delivery. Okay, so they have the cell body here containing the DNA, the genetic information of the male, and then a tail, and that's it. They don't have a lot of cytoplasm, they don't have a lot of structures, they don't have any nutrition stored with them. Their number one concern is to swim fast and deliver. And unlike the egg that has lots and lots of nutrition with it, they don't have anything to support it, which is why they need to be delivered in these secretions and as semen and not just sperm. Everything in the secretions is there to help. For example, the semen, those other two secretions from the seminal vesicles and from the prostate gland, have a lot of sugar, in this case fructose. And this is going to be energy for those sperm, nutrition for those sperm. They also have things that make them very alkaline or very basic. And I shouldn't say very, they're a little bit on the alkaline side, so 7.2 to 7.6. And being a little bit basic is going to allow it to neutralize the acidic conditions that it's going to meet in the vagina. The sperm need to be in a neutral or slightly basic environment. If they meet acid, they're going to die, so this helps protect them. Next is something called seminal plasmum. This is basically an antibiotic, so this is there to protect any bacteria the sperm might meet. And lastly, there's some enzymes. I don't expect you to know any specific names. Just know that they're there to do two things. First, they enhance the mobility of the sperm, so they kind of wake up the sperm and make them swim quickly. They also inhibit the female immune response. If you think back to when we talked about blood types, our body makes antibodies for things it doesn't recognize. It attacks things that it doesn't recognize as part of its own body. So when sperm enter into the female natural response would be to recognize it as something foreign, something that didn't come from them, and attack. And there's enzymes contained in the semen that prevent it from doing this. There's only about 2 to 5 milliliters of semen per ejaculation, so a very, very small amount, but the sperm are so tiny and they're produced in such great amounts that for every milliliter, there's about 50 to 150 million sperms. We're actually delivering quite a bit of sperm, and we only need one for actual fertilization. Okay, lastly, we're going to talk about the external genitalia. So this is the things that are located outside the body that are assisting with delivery that contain these internal things we've just talked about. There's two main parts. The scrotum first is the sac of skin that's actually going to contain the testes, and there's a couple reasons that it's external. Your body actually talks about it seems like a strange place to put something that's so important, something that's going to be creating your offspring, to put it outside the body where it's not very protective, but there's actually a very good reason. Okay, so this is a sac of skin that hangs outside the abdominal cavity. And the reason for this is that sperm need to be at a slightly lower temperature than most of our internal organs need to be. So if we put these testes inside the abdominal cavity, we'd actually kill all the sperm because it would be too warm for them. Okay, so typically the scrotum is kept below body temperature. Now, if temperatures drop and they need to be kept warmer, muscles can contract, and this does two things. One, it pulls the scrotum toward the body, so your natural body heat can keep it warm, but also all of that extra skin that was allowing it to hang lower is now wrinkled up, and it creates more surface area to keep the testes warmer. The second external structure is the penis, and this is designed to help with delivery of the sperm. So it contains, surrounding that urethra is several layers of erectile tissue, meaning that they can be relaxed most of the time, but then they can be designed to flood with circulation, flood with blood, and grow in size and length. And this is going to help then be able to deliver the sperm. There's three main regions of the penis. The first is the shaft, which is the length of the penis that surrounds the urethra as it's exiting. Then the glans penis is the head of the penis at the end, and then that's surrounded by an extra layer of skin called foreskin that may or may not be removed through circumcision. And when we talk about how ejaculation actually occurs, we'll talk a little bit more about these structures. Okay, that's going to be it for this lecture. So again, this matches page 546 to 549 in your textbook. Make sure you're familiar with all of these structures. So what are the actual structures of the testes? What is the pathway that they take out through the duct system? And some important things about that, such as why the epididymis is so convoluted and coiled, why there's smooth muscle in the vas deferens, why do we produce the different types of secretions that the three glands we talked about produced, and why, for example, the scrotum needs to be outside the abdominal cavity. So keep in mind that idea of structure being there for a reason. How does it help the function?